In this video, we will discuss the role of viruses in cancer and techniques utilized to cultivate viruses in the laboratory. Animal cell cycles are regulated by genes. The genetic controls dictate that some cells are not permitted to divide, while others that are permitted to divide are very closely regulated to prevent uncontrolled cell division. Any alterations or mutations to the genes that regulate the cell cycle can lead to uncontrolled cell division. Uncontrolled cell division is referred to as neoplasias, and a mass of cells that arise from uncontrolled cell division is known as a neoplastic tumor. Cancers that remain in one location are benign. Others invade surrounding tissues and may move to other locations within the body. These are known as malignant. The ability to move and invade is known as metastasis. Malignant forms are referred to as cancer. One particular type of gene is the proto-oncogene, which can be activated to abnormally function, thereby causing cancer. There are several factors that can activate oncogenes, including ultraviolet light, radiation, carcinogens, and viruses. Viruses have been associated with 20 to 25% of human cancers. Viruses that can cause cancer are known as oncoviruses. A feature shared by all oncoviruses is that they integrate their nucleic acid into the host cell's chromosome. Scientists need to cultivate viruses in order to study them to gain knowledge regarding their structural composition, their replication cycles, and effects on their host. Scientists also need to be able to identify viruses in clinical samples and use an understanding of the virus to synthesize antiviral drugs and immunizations. Because viruses are obligate intracellular parasites, early attempts to cultivate viruses were very limited. Scientists were restricted to growing the virus in the host organism. We will look at several ways to cultivate viruses in today's laboratories. Bacteriophages are cultivated in bacterial cells in the lab, a process that is relatively easy. Bacteriophages can be grown in bacterial cells that are suspended on liquid media or in bacteria within colonies on a solid media. To cultivate bacteriophages on a solid media, the bacteria and bacteriophage are mixed in a warm liquid medium containing agar. The medium is poured into a petri plate and allowed to cool and solidify. Following incubation, the microbiologist can locate the bacteriophage by looking for the presence of plaques. A plaque is a clear zone on the media where growth of bacterial cells is interrupted. The plaques are areas where the bacteriophage has lysed their host cells. Each plaque is representative of one bacteriophage in the original sample. Thus, microbiologists count the number of plaques to determine the number of phages in the original sample. Plant and animal viruses can be grown in plants and animals respectively. Living plants and animals are more expensive to utilize to study viruses and more difficult to maintain. A number of types of animals have been used to study viruses, including mice, rats, rabbits, guinea pigs, pigs, and primates. There are some viruses that cannot be studied in this fashion because they may be species-specific for the human. A prime example of such a virus is the human immunodeficiency virus. Scientists have developed alternative ways to culture and study viruses that are more easily maintained, less expensive, and allow them to study even those viruses in the laboratory that are specific for the human species. Embryonated eggs are a useful, inexpensive way to cultivate viruses in the laboratory. These eggs have been fertilized and thus contain a developing embryo. Embryonated eggs are somewhat like a cell culture, but with many different types of cells. Viruses can be injected into the different membranes to cultivate them. For example, the influenza virus can be injected into the amniotic membrane or the allantoic membrane, while the herpes virus can be injected into the chorioallantoic membrane. Eggs have been utilized to cultivate viral components for immunizations. In fact, the inactivated flu vaccine is cultivated in eggs. Cell cultures are a useful tool in the cultivation of viruses in the laboratory. A cell culture consists of cells that have been isolated from an organism and grown in or on a medium. Cell cultures were first utilized in 1949 to cultivate viruses. John Enders and his colleagues used human cell cultures to cultivate the poliovirus. Later, in 1951, at Johns Hopkins, cells taken from the cervical biopsy of Miss Henrietta Lacks were utilized to establish a cell culture. Miss Lacks died shortly after her biopsy, but her cells still continue to grow today in labs around the world in cell cultures known as the HeLa cell line. Cell cultures are classified as a primary cell culture or as a continuous cell culture. Primary cell cultures are established from plant, animal, or human tissues. The cells are isolated and grown on a medium with the appropriate nutrients and growth conditions. Primary cell cultures tend to die out after a few generations. One line, known as diploid cell cultures, are derived from embryonic tissues of the organism and can be maintained for approximately 100 generations. Human diploid cell cultures are widely used for studying viruses that are specific for human hosts. Continuous cell cultures are those derived from cancerous cells. These cell cultures can be maintained for an indefinite number of generations. The HeLa cell cultures are an example of a continuous cell culture.